So,
You guys saying there's a lot of A's here?
yeah, well, no, I'm saying that looks like a typical engineering thing. I remember some at Brown where like the A range went down to the high 30s.
Yeah.
I don't understand that. What's with engineering and the desire to make people cry?
I think the test designs are always bad. It's like just frustrating.
It's like if you can only answer one word of it and that's a B plus.
Yeah. It's like a miserable experience because you just feel like you're always behind and always
we have a great pleasure to have Patrick J with us today. Patrick is, as you know, and as stated here, is in the departments of Pediatrics and Genetics. And he graduated from the MD PhD combined degree program here at Loci, then went to Boston to continue his clinical training at Children's over there and continued his research at Harvard. And I've heard Patrick presenting several times, and I think he's really doing beautiful work on the development of the heart. What I was interested mostly was in the conduction system that he talked about, but today he'll talk about the genetic architecture of congenital heart disease.
Where do you want me to stand? Is he okay? Is he okay? Okay, thanks for inviting me. It's generally rare and kind of ironic that I get to speak in front of mostly engineers, because I often think that were I smarter, I would have become an engineer or a physicist. And so I think to make up for that, I spend most of my time thinking about the problems that I see, or I imagine how engineers and physicists might go about thinking about the heart as a machine, or thinking about gene regulation in mathematical terms, even though I don't understand machines and I really don't know much math. So what I'm going to talk to you today about is congenital heart disease. And just for orientation, basically what the bread and butter of what pediatric cardiologists deal with is plumbing. We also do some electrical work. And the plumbing, neither is that complicated. So this is a normal heart. Blue blood, which is deoxygenated, comes back from the head and the lower body to the right atrium. It goes to the right ventricle. The right ventricle pumps blood out to the lungs. The lungs put oxygen in the blood. They come back via the veins to the left atrium. It goes to the left ventricle. And then the blood gets pumped out again to the head and to the lower body. This is the basic plumbing. There are valves in between the atrium ventricles and between the ventricles and the arteries. Basically, any rearrangement or abnormality of the anatomy that you can imagine has probably been described in humans. So there can be holes between either the atria or the ventricles, so that blood goes from the left side to the right side, and hence too much blood flows to the lungs. This can be incredibly bad for babies that have large, what are called shunts. The lungs are like a sponge, a dry sponge, if you could look at them. And if too much blood flows to the lungs, they become sopping wet. It becomes very hard for babies to breathe. And unless they receive medical or surgical attention, these babies are going to die at about a month of age if the hole is really very big. If you have a large ASD, symptoms and bad sequelae do not develop until adulthood. So there are other types of defects. You could have normal 4 chambers with a wall in between, but there's something called a coarctation, where the blood does not flow from the aorta to the lower body. This develops at about 2 days of age in humans. In a mouse, probably about 6 hours of age. If you don't have blood flowing to the lower body, the body becomes acidotic, shock develops, and again, death ensues. You can have the arteries switched around. These are just examples of the bizarre things that we see. So blue blood comes back to the right heart and gets pumped out again to the aorta. The blood from the lungs goes round and round between the lungs, the left ventricle, and the lungs again. And what happens is, if this connection closes in the first couple days of life, basically no oxygenated blood gets out to the body, no deoxygenated blood goes to the lungs, and again, you have circulatory collapse. There are diseases in which chambers of the heart don't grow. So this is an example of hypoplastic left heart syndrome, where basically the left ventricle and the associated structures, the mitral and the aortic valves, are too small to sustain cardiac output to the body. And so what the cardiologists and surgeons have to do then is turn this into a series circulation where the right ventricle pumps blood to the body, and the lungs fill by passive drainage from the veins. Here's an example where a valve doesn't grow. So between the right atrium and right ventricle is the tricuspid valve, and instead of a nice valve that you can see, say for example here, you see this plate-like structure. So now the blood has to go from the right atrium to the left atrium out to the left ventricle, and somehow you need to design a system such that blood can get both to the lungs and to the body. It can get even more bizarre. So in this particular situation, the left and right ventricles are totally switched. So, you know, in these cartoons, it's hard to tell that the ventricles are actually different, but if you could open up the heart and look at it, you would see that the anatomy of the left ventricle, which is bullet-shaped, is very different than the right ventricle, which is more like a V shape with some thickness to it. The people who have this type of defect usually present in childhood, although occasionally you will actually find an old person who has this type of heart doing just fine. And here's an example where both atria drain into the left ventricle, and there's a rudimentary right ventricle. So these are the types of bizarre defects that we see. The other observation that I would like to tell you about relates to the story of a baby and her aunt. So this baby was diagnosed in utero with hypoplastic left heart syndrome. Basically, you can see here a normal baby's heart. You can see the right atrium, right ventricle, left atrium, left ventricle. You can see the valves in between. And here, as in the cartoon, the left ventricle is itty-bitty, and that's not going to sustain life. This is a very common story. So most of the children whom we see have no family history of congenital heart disease, which is kind of interesting, because if you ask a developmental biologist or a geneticist, they will tell you that probably all congenital heart disease results from genetic mutations. So there's a discrepancy between what we think about from working in the lab and what we see in real life. This baby was the first to young parents. This is an important fact because parents are always asked whenever their child has any serious disease, is this going to happen again to a subsequent child? And the answer is, usually we don't know, maybe. You know, that's really not a good answer, and for scientifically, you know, there's no mechanistic basis or thinking involved.
Patrick, that's a good question. So it could be spontaneous mutations arising which can cause very rare genetic diseases. So what is the correct answer what is the statistics say about the chance that a second child has it? Is it any different than normal?
In the next slide. So not much was thought about this baby until her aunt, who was 17 years old, presented with exercise intolerance, so she couldn't keep up. And what was discovered was that she had an atrial septal defect. So this echocardiographic picture shows blood flow by Doppler imaging of blood flow from the left atrium to the right atrium. This is during diastole, so you see it flowing all the way into the right ventricle. You can see in a close-up here that she has about a 1 to 1.5 centimeter defect between the left atrium and right atrium. This was closed in the catheterization lab with an umbrella-like device. And you can see in now this picture after catheterization that blood does not flow across the atrial septum anymore, and instead you see blood flow normally across the atrioventricular valves. So referring to Colin's question, this is the family tree. The first patient had hypoplastic left heart syndrome, which is a terrible disease. So, and in general, very difficult to fix. Even if we fix it, these children commonly have serious medical problems. In contrast, we find an aunt who has a relatively simple defect for us to fix. Once you fix it, in general, these patients do very well. So in general, the incidence of congenital heart disease in the population, if you examine all newborns that come to you, is roughly about 0.5 to 1%. 0.5% has significant congenital heart disease. By significant, the definition is that you need a cardiologist or a surgeon to survive. The probability, so most congenital heart disease, I told you, is sporadic. It's not obvious from talking to the family that there's a genetic basis to it. But if you do large surveys in populations, the probability of having an effective relative with congenital heart disease is greater than the general population incidence. It goes from about 2 to 10% depending upon the type of defect. And so you can infer then that at least in some patients, there's a genetic basis that's heritable. But maybe not in all. And so we examined and did echocardiograms on the siblings of this girl. They were all normal. These parents obviously want to know if their subsequent child is going to have a defect, so we can do fetal echocardiograms. This boy ultimately turned out to be normal. And obviously, I think you can understand that parents do not want to hear that the chance of the next child is 3%. You know, they just want a yes or no answer. And as scientists, we want to understand why is there this discrepancy between, say, clear-cut Mendelian genetics, where the probabilities are well understood, and this more vague epidemiologic observation.
And the parents are perfectly normal? They were echoed as well? Or not?
So I did not echo these parents. They were not available to me. So I can echo people who are below 18 or below. After that, it's not my problem.
But if you're trying to follow the genetics...
Yes, you're right. So they should get echoed. And they know.
I mean, I'm just wondering, how often is it undiagnosed that there is actually some congenital defect that you could see if you really did echo people, but it may never crop up? You know, it may never reach clinical assessment.
So severe heart defects are incredibly easy to diagnose because they come sick. Other types of defects, so that's the function of pediatricians, and that's why you send your child to the doctor on a regular basis, is because if they don't catch it the first time, they may catch it the second or third time. And that's
so the ASD at 17, you're still calling that pediatric?
Yes. And you know, that's what we're taught to do in schools, to listen to hearts and actually understand what the noises sound like.
But maybe the parents would never have been checked, right?
Well, yeah, so it's not a perfect system, you know. So, but the point of physical examination, it's like a genetic screen. That's how I think about physical examination. We're trying to catch abnormalities out there. We can catch most of them. Sometimes a good doctor can catch most of them. Sometimes you're going to miss them.
But I guess the question I'm asking is that there are things which the individual would not take themselves to a cardiologist, and if they didn't happen to make it through the system and be caught as pediatric cases, they could have congenital defects that have never been diagnosed. Is that true?
That's possible.
That's the point.
That's possible, yeah.
Okay.
It happens.
Obviously not the severe ones.
Even the mild ones happen. I think relatively frequently if you have a good medical system. But that's not important because we're interested in engineering and genetics. So what? Because you guys are engineers, I'm going to give you a project, and then I suspect that you guys will figure out how to design a very good pump for the masses. Basically engineer a system to do this, and figure it out without looking at thousands of mice, which my laboratory has done, and without using probably more than a dozen students and cardiology fellows to figure what nature has essentially figured out. So here are the design specifications. The vast majority of hearts in the population are normal, so more than 99%. You know, why? So that's the specification that we need. And why is that? I would assert to you that the reason is because of evolution. For a species to survive, you need a heart. You need a heart to basically get you past the newborn period. You need a heart to get you to reproductive age. And if your pump is poorly designed or subject to flaws, a species would die off. So that's one specification. Here's an observation that we make empirically. Most congenital heart disease appears sporadic, so probably that's a clue for how this machine is designed. Carriers of genetic mutations. So I think that the parents, even though I did not echo them, but we see this quite commonly even when we do, we can infer that they carry genetic mutations, but somehow they have escaped disease. And I think you can imagine then that if you were to engineer the design of a very complex pump, complex both in terms of the anatomy and in terms of the demands placed upon it, physiologically, there must be systems that check against perturbations towards its development and function. The last empiric observation is that presentations of congenital heart disease vary incredibly widely. They can go from simple holes in the heart to very bizarre forms where valves don't exist or chambers are totally gone. So in thinking about this problem, most scientists, at least molecular biologists, developmental biologists, and geneticists, certainly physicians, focus incredibly upon things that are abnormal. And we spend our time mostly wondering about why are things abnormal, why do people get disease? If you want to look at the big picture, I think you need to take a step back and realize, again, that the vast majority of us are normal. And it's quite banal. And the question is, why is that? So back before the high-powered molecular biology and genetics, a number of embryologists had wondered about this problem. One of them was Conrad Waddington, truly a Renaissance biologist. He was around in the 30s and 40s. He worked for the British Royal Air Force, and as part of his research for the Air Force, he developed algorithms for finding U-boats. And after World War II, he decided that genetics was the way to go to understand embryologic problems. And what he recognized, again, was basically that the norm is very common. Why is that? And he reasoned through a series of experiments that things like the number of bristles in the fly, which is highly invariant, must somehow be built into the mechanism of building the fly. A number of other investigators basically built on his experiments, and they showed that whisker number in the mouse was also highly invariant, and there seemed to be genetic genes that were involved in determining this highly invariant phenotype. If you look at the veins in a fly, the pattern is also highly invariant. On the other hand, when you mutate it, selected for mutants at least, there was more variability. But even when there was variability in these mutants that affected, say, fly bristle number or mouse whisker number, there were always, within these populations, a large fraction of normals. Among the abnormals, you could actually select for traits that led to, in successive generations, that led to either high or low whisker number. So what Waddington envisioned, even before genes were fully understood, was that development followed a process of canalization. So up here, at the top of this topographic map, sat a totipotent egg. As the egg divides, each cell makes decisions about which cell type or tissue it wants to make. And development, he reasoned, flowed down these pathways towards its lowest potential, which represented the wild type invariant form. Perturbations could push this cell onto another pathway, and underlying this topographic map were genes that somehow were hidden in the wild type population, and you would only see their effect in the presence of a major mutation. And so canalization as an idea basically fell by the wayside after some very elegant experiments in the 40s and 50s, because they really couldn't push it much farther. But now I think with the advent of the Human Genome Project and certainly advances in mouse genetics, we're now able, I think, to dissect the underlying genetic basis of this complex phenomenon. Here's an example, an elegant example from Greg Gibson's laboratory. He spent his career basically trying to figure out various phenotypes in the fly and why they are canalized. This is a fly eye. This is a normal fly eye. In the fly eye, it's a compound eye. There are about 800 ommatidia. The ommatidia are composed of 8 photoreceptor cells, 4 cone cells, and I think 3 pigment cells. It's highly invariant pattern. Mutations of 2 different genes, it's not important for the purpose of this discussion, can affect the quality of the fly eye. And what they did in a very simple experiment was basically grade the roughness of the fly eye after breeding these mutations into wild-type fly strains collected from vineyards around the world. So what scientists, postdocs, and they actually had one undergraduate student go to the Ann Arbor fruit market to collect wild-type flies, did was breed these genetic mutations into strains from California, France, Italy, Africa, and quantified the roughness of the eye, with 1 being totally normal fly eye and 10 being totally abnormal. And what you can see here, so ellipse and 7-less are 2 different genes, is that depending on where the flies come from in the world, a gene that's thought to be incredibly important in a laboratory experiment can have basically no effect in one particular population, whereas in other populations it can have a great effect. And then when you look at a separate gene, fly eyes here, which are normal, can be totally abnormal up here. So there seems to be no correlation between the wild-type genetic background and how severe a fly eye phenotype is, or with the particular gene involved. And you might wonder why this is. So, closer to the heart, you might wonder whether cardiac development is itself canalized. Down syndrome is a disease in which a person gets 3 copies of chromosome 21. So that's a serious genetic perturbation. And when I ask cardiologists and pediatricians what the most common cardiac phenotype is in Down syndrome, which is commonly associated with heart defects, they will say AV canal defects, and I will tell them wrong. So AV canal defects represents only about 1/4 of the heart defects we see in these children. Simple septal defects and another defect called tetralogy of Fallot represent the other 1/4. But more than half have totally normal hearts, even though they have 3 copies of chromosome 21. So again, you get this idea that there's something else going on. The embryo can take huge hits to its genes, or take huge environmental perturbations, and somehow figure out how to correct itself. Or you might imagine that there are mechanisms that allow these animals to correct themselves and get back on track. So how does that happen? What we need to do then, instead of speculation about these observations of the world, is bring it into a laboratory model. And so the gene I work on, NKX25, was the first gene discovered, at least in humans, to cause congenital heart defects when mutated. It's the homologue of a gene in Drosophila called Tinman, so named because knockout of this gene in the fly causes absence of the heart to develop. Tinman and NKX25 have relatively similar patterns of expression in the embryo. So in the fly and in the chick, which is the same as mammals, you see expression of Tinman or NKX in the lateral mesoderm. These cells, during development, come together to form the heart tube. And then in birds and mammals, the heart tube loops and ultimately forms the 4-chambered heart that we're used to. So the idea back in the late 80s was that maybe mammals had a master gene that was responsible for cardiac development, as in fly, and if we knocked it out, the heart would disappear. And I think it was a little bit disappointing to the 2 labs that made this, when they found that when they knocked out NKX25, the heart did form. So here's a picture of the heart here in a null mutant, but it wasn't exactly normal. So what you can see here in the wild type is at day 9.5 of gestation in the mouse, is that you have the beginnings of the atrium and ventricle. You can see a nice atrioventricular canal, so the atrium and ventricle are clearly demarcated. Similarly, the left and right ventricle, you can see the beginnings of them. In the mutant, you just see this ill-defined heart tube where you don't have clear demarcation of the atrium and ventricles. You see a single bulbous ventricle. When you section it, in the wild type, you see the beginnings of the 4 chambers of the heart. In the knockout mutant, you just see the atrium and ventricle. This mutant stops growing in about day 9.5 because it reaches the limits of diffusion, oxygen diffusion. So after day 9 of gestation, you need a functioning circulation to pump nutrients to the body. And so that's why this embryo dies early in gestation. Work on this animal would have stopped were it not for the discovery soon afterward of mutations of NKX2-5 in man. So cardiologists from around the country had collected these families, and they had noticed that in these families there was a high incidence of heritable atrial septal defects associated with atrioventricular block. We won't talk about the mechanism of baby block today. We have figured that out. And so in traditional genetic linkage analysis, every black person who has a black circular square was discovered to have an NKX2-5 mutation that causes loss of function. And then the question was, in the heterozygous animals, do they get heart defects? And so what we did was basically look. So I had told you before that heart defects are bad, and certain types of heart defects will cause death in children, either at roughly 2 days or 1 month of age, depending upon changes, which depend upon the changes in going from a fetal circulation, where you don't send blood to the lungs, to the postnatal circulation, where you do. What we discovered was that mice in one particular strain background, we'll talk about what strain backgrounds are in a second, do develop heart defects. So this is an example of an atrial septal defect, where you can see blood basically flowing freely from the left atrium to the left ventricle, from the left atrium to the right atrium. Here's an example of a muscular ventricular septal defect, where there's a hole in the ventricular septum and blood flows from here to here. This is a different type of ventricular septal defect. This is actually the most common type of defect that we see in man. It's called a perimembranous VSD, and it sits right underneath the aortic valve, between the aortic valve and the tricuspid valve here. And you can imagine that it results, at least in part, from this ventricular septum not growing up all the way from the apex to the base of the heart. So by tallying the genotypes in the notebook, we realized that about 15 to 20% of our animals were missing when we separated them from their mothers, largely due to these types of defects, these large shunts that make it hard for them to breathe. So to understand the rest of the talk, it's important to understand what inbred mouse strains are. Usually I don't talk about, but I think for you guys, for the engineers and physicists in the group, it's important to understand the significance of inbred strains. So humans are genetically heterogeneous. I think we all appreciate that we all have a mixture of genes. No two humans are exactly alike. In contrast, I can go to a website and buy a mouse, a strain of mouse, where every mouse within that particular strain is genetically identical to every other mouse within that strain. For that, everyone has 2 copies of a gene or a chromosome. Those 2 copies within a particular strain are identical. That means from each parent, they basically get the same genes. Basically, everyone is an identical twin. Where inbred strains come from, basically, it came from hundreds of years of propagation of what were called fancy mice. So, about 500 years ago, mice were bred for pure coat color or traits like what was called waltzing. These mice had vertigo, and they would spin around. And so this was a hobby of royalty and wealthy people. Abby Lathrop was a farmer in western Massachusetts who basically failed as a farmer, but realized that she could make a living by collecting these fancy mice and selling them to hobbyists. And then a group at Harvard at the turn of the last century realized that they could use these mice to test principles of genetics. So at the time, Mendel's laws were rediscovered, and the question in the late 1800s and early 1900s was whether the laws discovered in peas and probably insects, whether they applied to higher organisms like mammals and birds. The competing theory at the time was something called blending inheritance, where, say, for example, you had a short mother and tall father, the child in between, the child of them would have medium height. And it was not obvious at the time whether this was from blending, ill-defined blending of traits, or from inheritance of classic Mendelian genes. And so they could take these mice that were black, white, whatever, and breed them to figure out the genetic basis of various traits. So what we did was take NKX25 mice that had been bred into the C57 black 6 background, similar to the experiment that Greg Gibson had done, breeding a mutation into a wild-type fly strain collected from anywhere in the world. And we crossed them to mice, white mice that were called FBBN. So the progeny here are basically half black, half white. None of these mice had heart defects, we noticed. And then you might wonder then whether it's because there's a gene here in the background that increases susceptibility to heart defects in the presence of NKX25 mutation, or if there were protective genes, or there was something more complicated. And the way to figure that out is to take this mouse, breed it against the parents, or amongst themselves, and then see whether these mice have heart defects. Now, this mouse is normal. All of these mice had heart defects, whether you breed them to the parental strains or between themselves. So it suggests then that the simple model that the black 6 has some susceptibility gene only is incorrect. So there must also be susceptibility genes in FBBN, as well as protective genes here. We did the same experiment for crossing them to AJ and got similar results. So this phenomenon where the F1, the first generation, looks normal is called heterosis. Heterosis is the phenomenon that keeps companies like Monsanto in business. So farmers buy corn from Monsanto that are basically F1 hybrids of 2 inbred strains of corn. Monsanto can sell these corn because the F1 has, for farmers, a more desirable trait than either of the parental, than can be predicted from either of the parental strains. In this case, the desirable trait for me is the absence of congenital heart disease. Farmers cannot take their seed corn and bypass Monsanto. Basically, they're stuck. They always have to go back to the company, because if they breed their seed corn amongst themselves, they're going to get basically corn that has a mixture of phenotypes, and they won't have as consistent a trait, whether it's high yield or high protein in their corn. So, and that's the analogous situation here, where we get heart defects in our second generation animals. So to do this type of genetics and to find the genes that underlie the development of heart defects in the presence of NKX25 mutation, we need to screen a lot of animals. And so what we do is we take mouse hearts, we put them, we collect them every morning, 365 days a year. We embed the hearts in blocks of wax, section them as you would a CT or MRI scan, and we look at them under a microscope. And in these pictures, you can see basically everything in the heart. So you can see the ventricles, you can see the aorta and pulmonary artery, you can even see, let's see, right here. So you can see little things like the left coronary artery coming off the aorta. We can see little VSDs under high magnification. So basically holes the size of a red blood cell, which is about 10 microns in diameter. This data shows what I just explained to you qualitatively. So in the C57 background, mice that are heterozygous mutants for NKX25 have a high incidence of either ASDs or VSDs. When we cross them to the F1 generation, the incidence of defects is either low or nil. Wild type mice rarely have heart defects. What we want to find then are the genes that influence the development of heart defects in the presence of mutation. So this is linkage analysis explained very simply. You have to dig back to your high school biology. What we're looking for, each square represents a region of the genome. What we're looking for are genes that are associated with the development of heart defects. So all these mice have a heart defect, and we can differentiate between the white mouse and the black mouse genes. We're looking for a mouse, we're looking for genes that basically little v's in every mouse that have a heart defect instead of big V. Does that make sense? And so here is where we do, that was the simple case. In reality, this is a complex trait. Complex traits are traits or diseases in which many different variables influence the development of phenotype. And what we did here is we took all the F1 animals that have been crossed among themselves, that have MKX25 mutation, and we divide them into two groups. So mice whose hearts are totally stone cold normal, and mice that have a perimembranous VSD. And then we have markers that differentiate whether a particular segment of the chromosome came from either the black or the white mouse strain. And we will look for statistical association of that particular what's called allele with the presence or absence of a defect. Very simply, basically any score above 3 means that we found a gene in this particular region of the chromosome that's associated with the presence of a VSD and the presence of MKX25 mutation. As you can see, we found a number of them that are above 3 and a number that are close to 3. So it's more than one gene that is involved in influencing the development of the heart defect. Here's an example of the risk associated with having a particular genotype at a location on chromosome 10. So in this spot, which is near the centromere of chromosome 10, if a mouse has 2 copies of this locus, it has a high probability of getting a VSD. If it has just 1 copy of the white mouse gene's version, white mouse's version, the risk is reduced substantially. And we know that at this locus, this gene is dominant because it's the same whether you have 1 or 2 copies. And you could build similar risk plots for all the other loci. So I told you that there are 2 different types, at least 2 different types of VSDs, muscular and membranous. We can build the same genetic linkage analysis for muscular VSDs. And basically what this shows is that the genetic pathways that lead to one particular type of defect or another is not exactly the same. So to physicians and to most developmental biologists, the way thinking is now is if you have a mutation of a major gene, you get this range of defects. And we don't know why, that's just the way it is. But it would be nice, you can imagine, if you understood the pathways that led to one defect or another, to be able to manipulate that and basically, and hopefully steer what could be a severe defect to a normal one by nudging the activity of one of these genes. So here, what we see is the map, the linkage map for muscular VSDs. We see that some loci are unique to muscular VSDs, so suggesting that there are 2 different genes that lead to one defect or the other. There are some loci that are shared with perimembranous VSD. So now we're setting up a genetic hierarchy. The mutation of NKX25. If you have a polymorphism of gene A, it could lead you to a particular type of to one of 2 types of defects. And then if you have below that gene, you can steer your heart towards one type of defect or another. In a separate cross to the age A strain, we get a qualitatively similar pattern of genes. So we find a number of genes that are significantly associated with the development of VSDs. Some of them appear unique to this particular cross. Others appear to be shared with the FEBN cross. So what you can see here now is it's not just 1 or 3 or 4 genes that steer the heart towards a particular type of VSDs. Every time we look at a new cross, we see more genes. So it's a lot of genes involved in steering the heart towards or away from a defect in the presence of a serious mutation. For the geneticist, they always want to know what genes underlie these chromosomal locations. And so with the advent of the Mouse Genome Project and sequencing of inbred strains, it's relatively now simple for us to finely divide these large peaks, which can represent hundreds of genes, into smaller intervals. So I told you that mice derive from strains that were propagated by hobbyists. So most mice used in the laboratory come from probably just 4 different types of wild-type mice that you find out in the field somewhere. And you can basically trace the genetic architecture of these inbred strains to these 4 genes. And so what we're looking for depends upon, where the genes lie depend upon how we've mapped it. So if I know that C57 has a gene that increases risk that's not found in FEB or AJ, I want to look at intervals that are different but shared between these two. Whereas if I find a peak that's, say, unique to FEB, the FEB cross but not AJ, I would look at a place like this. And you can see how a very big region can be winnowed down into smaller regions where we can find genes of interest. And what this is the genetic term for this is finding things that are identical by descent. So somewhere hundreds of years ago is a mouse, the great-great-great-great-grandfather, that passed on its genes to C57 or AJ or FEB and AJ. And what we want to do then is to find those genes. And what we've done is basically pool the data from the 2 crosses, the crosses to FEB and AJ. I told you that mod scores of 3 are significant. But if a gene is identical by descent, basically we've used the power of hundreds of years of what's called recombination to really focus on intervals that are important. And so these 2 loci pop up and become highly significant. So this is basically a log score. So this is the probability of this is 1 in 1,000. So this would be 10 to the 5th. That's like 1 in 10,000 of something real sitting there. We find, I told you that in the real world, we find heart defects that are incredibly bizarre. We've now studied, I think, more than 5,000 mice. I think we're probably approaching 6,000. And we're beginning to find not just common defects like atrial and ventricular septal defects, but rare defects. Here's an example of a common atrial ventricular canal, which is frequently seen in children with Down syndrome. So in a common atrial ventricular canal, they basically have, the simple way to think about it is that they have 1 valve between their atria and ventricle. So it looks like a fish heart. And we find that it's more common in the AJ cross than the FEB, suggesting that there are genetic polymorphisms that influence not just the development of common defects, but of rare defects as well. These data, put in bar graph form, shows this particular pattern. So we're beginning to reproduce the epidemiologic pattern of heart disease. VSDs are more common than ASDs, are more common than AV canal, and we've seen a rare defect called double outlet right ventricle where both arteries come off the right ventricle. Genetic modifiers appear to play a very important role in the types of heart defects that we see. Even though every mouse that we study has an MKX25 mutation, not every mouse is the same. And I think you can begin to understand the variability that we see in human populations. So ASDs are more common in the AJ intercross than in the FEB intercross. AV canal defects, although relatively rare, are significantly more common in this particular cross. Another thing that we realized that we could do in trying to sort out the genetic hierarchy of heart disease, and for you to figure out the engineering problem, is you might imagine that we have one gene controlling the development of holes between the ventricles. You might ask whether these other genes influence development of defects unrelated to the ventricles. So one of the problems in developmental biology now is that even though we've found probably 3 or 4 dozen genes that are implicated in the development of congenital heart defects, so holes in the heart or abnormal plumbing, there's really no underlying logic at all in why a particular mutation leads to a particular type of defect. There's no hierarchy, kind of in the same way that the cancer biologists have a hierarchy where they can say, we need this series of mutations to lead to colon cancer or to lung cancer or whatever. In heart disease, it appears totally random. And I think as engineers or physicists, you must understand that cannot be the case. So we ask a very simple question. Are there genes shared between parts of the heart that, as far as we can tell, are totally different? And they're not tied together in any developmental pathway that we know of. The way we approach this problem was statistically. So if there are genes that are involved, you might then that influence the development of 2 disparate types of defects. You might imagine that the probability of a heart having 2 defects is higher than the product of the separate incidence of the two defects. Does that make sense? So basically, if two defects occurred entirely independently, the incidence of, say, having both an ASD and a VSD would be equal to the red bars. So it's just the product of the separate incidences. What we see in reality is that we see many more hearts. We see more hearts than expected in all our different crosses that would be accepted purely by chance, suggesting that there are some genes involved. And so to go about that, what we want to do then is to look at the ASD map. This is something that we couldn't do in a conventional developmental biology experiment, where usually you look at just a couple dozen animals. So to get this type of data, we need to look at hundreds. And so an undergraduate student in that, Ashley Luca, has actually done this. And what we found now is that there do exist some genetic loci that seem unique to ASDs, but there are also some that are shared with ventricular septal defects. We've done so many mice that we're beginning to recapitulate human epidemiologic patterns of congenital heart disease. So Colin is asking the very important question, what causes heart defects? So there could be heritable mutations. There could be spontaneous mutations, genetic mutations. There could be random environmental perturbations. So, say your mother is a diabetic. Children of diabetic mothers have a slightly increased risk of congenital heart disease. We've done so many mice now in controlled circumstances. I'm pretty sure that environment does not play a big role as far as I can tell. Genes, I think, play a big role. And it's genes that we can't see. So it's not NKX25. It's genes in the background that don't appear to affect the wild type form, that only manifest their effect in the presence of a major mutation. So in human populations, it doesn't matter where you look, whether it's in New England, Baltimore, Washington, the Minnesota area, Scandinavia. These studies have been done over different decades and with different populations. Wherever you look, the answer is always the same. VSDs are most common, followed by ASDs, followed by tetralogy of flow and these rare or bizarre defects. 0.5% of the population, like I told you, has significant heart disease. 1% have some abnormality that may or may not be serious. We see the same problem, same defects in mice in roughly the same distribution. And by inference, it suggests then that modifier genes contribute significantly to these epidemiologic patterns of heart disease. So on the x-axis here is the incidence per million of heart defects in man. And on the y-axis is incidence per 1,000 in our mouse experiment. You can see this distribution that's almost linear, where in particular strains, some strains appear to be a little bit more susceptible to certain types of defects. You can imagine the same thing going on in humans. So there might actually be just a few things that are the main causes of congenital heart disease, but many things that influence phenotype. And it might explain why human geneticists have had such a hard time finding new mutations after, in the 90s, what was a very productive period in finding defects in humans. So, it's possible that maybe we found all there is and everything else can be explained, in terms of variability, can be explained by modifier genes. So here is the engineering problem and how, after looking at all these data, my lab is beginning to put it together. You can imagine congenital heart disease or any complex disease in general. So I think this story is not limited to whole heart. It's probably things like diabetes, coronary artery disease, basically things that have been tough for the geneticist and human biologists to explain simply. Might be thought of in these terms, in very simplistic, mechanical terms. The vast majority of us sit in the middle. We're all normal. Disease exists at the ends. Something hits the embryo or the adult and pushes you off course. So, it could be an environmental factor. It could be a genetic factor. if you have in a population a heterogeneous mix of modifier genes, most of us are going to get buffered back to normal. So you get, you take the initial hit. Hits may be common, hits may be rare, but most of us will get back to normal. And this kind of makes sense from an evolutionary perspective. So if you're going to build a system, my task to the engineers was not to build a perfect machine. You wanted a machine that worked most of the time, so that a pump that worked most of the time, so that the population could live to reproduce. So, and that's why it's important to have a mixture of modifiers, and a bunch of them, so that just randomly, most people would get back to the middle. For this mechanism to work, however, you need genes that go both ways, because a priori, you do not know which way this cosmic ray is going to push you, whether it's going to push you this way or that way. So you might ask, why don't you just get rid of the modifier genes that push you off in this direction? In the lab, that would be a very short-sighted view. In the laboratory, I only know about NKX25 mutation. In the world, there may be zillions of other genes, zillions of other teratogens that can push an embryonic heart off course. And so what happens then is that a fraction of the population gets dealt the equivalent of a genetic royal flush. For most people to get back to normal, just statistically, some people are going to get a distribution of modifiers that once they get that genetic insult, these people will careen off course, going off to what looks like a bizarre abnormal heart. So I think then that you can understand how to engineer, mass produce a very complex, demanding pump. By having a robust design, we ensure that the vast majority of us are normal. And the way you ensure that is not to have one knob that you can dial in either direction, so that when the heart gets pushed, you can dial it back in. You need a bunch of different knobs, all of which have small effect. And the reason why is because the embryo or the individual has no idea when that perturbation is going to hit it, and it has no idea which direction it's going to push it in. So you need a bunch of little genes that get you back on track, so that most of us will survive. So what this gets at, I'm not an engineer, but I suspect that when the boss gives you a project, he or she sets up tolerances. So we want things to work most of the time, but we will tolerate a certain error rate. And that's where disease comes from. So a fraction of individuals will not be buffered against a particular insult, even though most people would be buffered against most insults. And then the combinations of the polymorphisms that you have in the background that do not influence the wild type form normally will then direct, in the presence of a major mutation, the type of heart defect that you get. And alleles, so versions of these modifier genes that might be protective in one particular insult, may be totally detrimental in the face of a different type of insult. So the work in this lab, in looking at thousands of mice involved, a small army of people listed here. Julia Winston is the graduate student who's done all of the genetic linkage analysis. Jonathan Courtney and Sook are the technicians who have sectioned the mouse hearts. It's incredibly labor intensive. Ashley is the undergraduate who I talked to you about. Bob Ashish and Martin LePage were cardiology fellows early on who helped us look at all the hearts. In case you're interested, we don't need cardiology fellows or even medical students anymore. We've gotten so good at this and done it so many times that I can take a high school student who's never had an anatomy class, and within a month, they can diagnose complex congenital heart disease. If you look at enough, you can recognize anything.
Only in the mouse.
In the mouse. No.
Only in the mouse.
No, I have taken my technicians, so the technicians look at every heart, into the echo lab at Children's Hospital. And so these fuzzy gray images, they recognize what's going on because they've seen so many. Jim Chevroux is a world-famous mouse geneticist who's really helped me guide my thinking in the lab. These are the people who pay for the work.
Thank you very much.
You got another one at the other end. Take this for
go ahead. I've been, yeah.
Okay. On this, going back and forth on the, your Gaussian peak there, going either bad or good, I think I think what I've gotten the impression from reading is that the general consensus is that by positive selection over many, many generations, we've got a good set of genes. Any change in that good set of genes is most likely to be detrimental. But on occasion, you will get one that is of advantage. And then if it's a real advantage in the environment, it tends to expand through the population. So that you have a high probability of, with a random mutation, of going the wrong way, and you have a small probability of going the correct way. So that those changes, there's a big difference in the probability going one way versus the other way. So where you go and get a bunch of steps that's detrimental, that's a higher probability than getting advantageous mutations. Just go ahead. Oh, you got any comments on that?
That's a really interesting question. For modifier genes, the definition of a modifier is a gene that has no effect on phenotype. So the phenotype that we look at here, or the fitness trait that we look at here, is a structurally normal heart. So these modifiers have absolutely no effect as far as we can tell. So they're not really positive or negative, and you might simplistically say that they would not be selected for in the population. They would only be selected for them are basically genetic mutations that basically maintain these polymorphisms in the population, right? Does that make sense? This actually leads to a very important question that we've thought a lot about in the past year, is where do these polymorphisms come from? And I've begun to appreciate that what evolution really means. So all biology, I suspect now, is based on some evolutionary principle. And I think the evolutionary principle that we're hitting on here that led to these polymorphisms of modifier genes relates to how the mammalian or avian four-chambered heart developed. So reptiles and amphibians have ASDs and VSDs. I imagine that hundreds of millions of years ago, maybe not even that long, a population of, say, alligators was selected to close its ventricular septal defect. So this particular population might have gone to high altitude where the oxygen concentration was low, or it might have had to run far and fast. So the heart would have to become more mechanically efficient. And so it would select in that particular population polymorphisms that cause that ventricular septum to grow all the way to the base of the heart and close the perimembranous VSD that it has. And so what's polymorphisms that in us are considered normal, might have been selected for in that alligator population. And I wonder if these defects that are associated with susceptibility are actually represent a type of aversion back to a more primitive allele. It might explain also the types of heart defects that we see, because VSDs and ASDs are seen in our most closely related vertebrate ancestors. AV canal defects, which are the next most common, basically look like fish hearts. And I wonder, if fish hearts are not more common because those particular polymorphisms have had more time to be selected out for whatever reason. It's purely speculation.
So your idea is that it just hasn't caught up yet?
Totally.
You guys saying there's a lot of A's here?
Yeah, well, no, I'm sorry, we decided to make people cry.
I think the test design.
As is stated here, is in the Department of Pediatrics and Genetics, and he graduated there and continued his research on disease.
It's generally rare and kind of ironic. Thinking about the problems that I see, I imagine. So what I'm going to talk to you today about is to do some electrical work. And the plumbing, neither is that complicated. And in the blood, they come back via the veins to the left atrium. Referring to Colin's question, this is the family tree. Our first in contrast greater than the general population incidence. It goes from about 2 to 10%. They were not available to me. So I can echo. They should get echo, and they know.
Crop up. You know, it may never reach clinical.
Assessment on a regular basis is because if they don't catch it the first time, they may.
But maybe the parents would never have been.
You're going to miss them.
But I guess the question I'm asking is that there are things which the individual.
Even the mild ones happen. So closer to the heart, you might wonder whether cardiac development is a disease in which a person gets 3 patients. And when I ask cardiologists and pediatricians what commonly associated with heart defects, they will say canal defects represents only about a quarter of the heart defects we see. Metrology of flow represent the other quarter, but the 3 copies of chromosome 21. So again, you get this idea, or take huge, or you might imagine that there are mechanisms that allow these animals to do. That was the simple case. In reality, this is a complex whose hearts are totally stone cold normal defect. Very simply, basically any influencing the development of the heart defect. Here's an example of 2 copies of this. The risk is reduced substantially, and we know that at this locus, this gene is dominant. Where usually you look at just a couple exist some genetic loci that in terms of variability can be explained by modifier genes. Guide my thinking in the way. These are the people who pay for the work. Basically, genetic mutations that basically maintain. So that's absence of so you would say that
Stillborn babies, do you think, are dying because of congenital heart disease? Now I have a question. So
my study section hates. So we find
how tight are your peaks? I mean,
so the